I'd love to show you some of the work that we do here and point out some of the things that we're working on now.
Okay, let's do this.
So this is the tissue culture room. This is where we do all of our sterile cell culture. And what we study actually.
Can I touch anything under there?
Just kidding. I probably wouldn't. Just kidding. What we study here in the lab are cells that come from cancer patients. So one thing that some of my patient families ask me is there anything that I can do to contribute to the research that you're doing? And I would say even just donating tissue samples or leftover biopsy samples, there is a ton that we can learn from each of those things. So over in this cabinet, we have perfect temperature control and oxygen control. And some of these will look familiar from biology classes in high school or middle school, but each of these dishes contains a different set of rhabdomyosarcoma cells that's grown. And obviously, my goal is not to grow them, but to kill them. But we think that by having all of these systems here, we can start dissecting which genes are important, which proteins are important, and then start moving into more translational studies using mouse models, and then eventually, hopefully, transitioning to the clinic.
How long does that process take?
That's a great question. So from the time that we identify a gene target to the time that we can create a drug and get it into patients, probably one of the fastest examples of that we've seen in recent years was in lung cancer. And I think that took about 10 years. And 10 years seems like a long time, but when you consider how long just running those clinical trials can take, I would say that we're on the right track. And my hope is that amount of time is just going to keep going down too. So this is an example of a sarcoma cell line. And when we look under the microscope, we can see all sorts of growth patterns and sort of the shape of the cells that fits with what we know about them. But more importantly, we can take these cells and then start either screening them against individual drugs, so asking which drugs are the most effective or the least effective, or starting to take individual genes and inactivating them with genome editing or gene editing. And again, asking which genes are the most important for this group of sarcoma cells to grow or not. Our hope is to then take that information and sort of spread that out to a bunch of other different cell lines and start finding patterns. So for instance, the pattern that we found was that rhabdomyosarcoma cells really need these chaperones to grow. We think we can actually learn a lot more by applying this methodology more broadly.
And you can see that under the scope.
We can see a lot of it under the scope. Some things we can't see under the scope. So for instance, the whether a cell lives or dies is one interesting thing to study, but we think we're probably missing a lot just by looking at that. Another thing that we do is sort of read the instruction book of the cell, and that's using something called RNA sequencing, which I remember reading about during my first year of fellowship. There was a brand new paper that was describing this crazy new technology, and now we don't even need to think twice about it. We just sort of submit it, and we know exactly what each of these cells is thinking and trying to do over a short time period, and that tells us a lot about which drugs are going to be effective or which combinations might be effective. And that's not something that we could do 10 or 15 years ago.
Can we see where that is here?
Absolutely.
Terrific.
So we're here in the Center for Advanced Technology at UCSF, which I think of as sort of like a dream garage. So every weird piece of equipment that you're staring at here is something that's relatively new or under development and constantly under use from all of the different labs that's here. And this guy that kind of looks like a beer fridge, the more that I think about it, is called a HiSeq 4000. And this is the piece of technology that we use to basically read the DNA or the RNA, so either the recipe book or the individual instructions that every single cell in our body or every single cell that we study in the laboratory uses. And this piece of equipment allows us to sort of get all that information back in maybe a day or two, whereas previously it took years and years to sequence that amount of DNA.
Fantastic. So then this information, how then do you actually make an impact upon the patient's DNA to actually convert that into a therapy? What happens then? You hear about this and gene therapy, but then what do you do? You give gene therapy, but how do you go from the beer fridge to helping a patient?
That's a great question. So, a lot of people are doing what's called mutation discovery or mutation analysis, and they're actually looking at patient samples and trying to find new mutations or new gene changes, and then sort of work backwards to figure out how important those are to the cancer. What we're doing is a little bit different. We're actually generating changes in the cancer cells through gene editing. And then using this machine to read out and say, okay, if we changed gene number 1 or gene number 2 or gene number 3? Which of those genes did we need to futz with in order to get the cancer cell to commit suicide or stop growing or whatever? And then once we figured out each of those genetic dependencies, we call them, so the genes that those cancer cells need, we can take a step back and ask, what drugs can we use to do the same thing that we were doing with our gene editing? And that's the process by which we can take this kind of big instruction manual of, the cancer cell sees this and does that, and really turn it into, this drug is going to work for this cancer.
Now, is futzing a technical term?
Futzing is absolutely a technical term.
Okay.
I had to practice my futz technique actually for a few years before they'd let me anywhere near this thing.
Is there like a certification for that?
Oh yeah, I'm a certified futzer.
Oh, it's good to know we're in good hands. So what else is in the lab?
Let's go and take a look at some of the other things that we're doing.
Okay, this has been a great walkthrough. Thank you so much. We actually we're here. There's obviously a great big giant lab. There's not time to talk about every station, but it's very clear that all over the place, in every corner, pretty much, in across the hall and then down the way, there's research in different shapes and forms going on all over the place, and it's really very exciting to see it and to have a chance to kind of experience that. Thanks for showing us, and I appreciate your time so much and everything you do.
Thank you very much. I think that the motivation to be here is really clear for everyone who is here. We feel like we're sort of pushing things forward, and even though it may seem sort of abstract when you're just looking at test tubes and things like that, I think at the end of every single day I know exactly why we're here, and that's to beat rhabdomyosarcoma. And thank you for helping support us in that journey.
No, it's my pleasure. Thank you so much.